The data from the Gallium study was very impressive. The Gallium study was mainly focusing on the treatment of combining obinutuzumab with various chemo backbones, bendamustine, CHOP or CVP, and it was being compared to conventional R chemo, which was again bendamustine R, CHOP or RCVP. And based on the follow-up data, at a median follow-up of almost 4 years, the PFS seemed to be very impressive in that there was improved PFS in patients who got G chemo over R chemo, and that held good in all the subtypes, but more so in the G benda versus R benda. However, one thing to note is the toxicity profile. There was definitely an increased incidence of both grade 3 to 5 adverse effects as well as serious adverse effects noted more in the G chemo arm compared to the R chemo arm. The acute toxicities such as cytopenias, neutropenia, febrile neutropenia or thrombocytopenia was more driven by the R-CHOP and R-CVP of the R chemo, G-CHOP and G-CVP of the G chemo backbone. However, the long-term toxicity such as infections and neoplasm, secondary neoplasms was mostly driven by the G benda and R benda. I think this is exciting time in the treatment of follicular lymphoma in the frontline setting, because for a long time we were just using the conventional anti-CD20 monoclonal antibody with other backbones. I think incorporating this novel anti-CD20 monoclonal antibody, which has more specific effects, I think that would be really exciting.